Good morning. I'm Rick Morgan. I'm the Chief Scientific Officer of Biopharma, and I'm going to talk to you about B-Bio this morning. So we're talking about B-cell medicines. It's a new and transformative type of cellular therapy, which has a number of unique properties. We produce proteins at durable and constant PK. They can be redosed and they're titratable. Significantly, there's no conditioning required for these cells to engraft. We've been able to show that you can produce essentially any payload from this platform, and they lend themselves nicely to an allogeneic approach. This is our team. Joanne Smith-Farrell is our chief executive. She just was elected to the board of the ARM, so we're very happy to that, and she'll be joining the ARM board in January. We have a very experienced team in cell and gene therapy, veteran leadership team. We are located in Kendall Square, Cambridge, Massachusetts, have a large facility there, both laboratory and CMC space. I'm going to tell you about our portfolio, which includes two lead products, B101 in hemophilia, B102 in hypophosphatasia. We are well funded. We closed a $92 million C round last year, led by Arch, Atlas, RA Capital, and Nextech, as well as others. So there are two programs in our clinical stage portfolio, B101. That program got from concept to IND in less than 2.5 years. As I'll describe, it really has the best TPP for any program in hemophilia B therapy. Our phase 1 trial is open in recruiting patients, and patients already been treated. And this is actually a large and increasing market. B102, next program, DC, developed this year in less than 15 months. Again, best-in-class TTP. It's actually a very complex protein, which we used AI to help design it, which turned out to be very productive. And we believe this will shift the paradigm of how patients are treated for hypophosphatasia. B-cell medicines. Okay. So B-cell medicines, BCMs for short, really combine the best of both worlds. It's a robust ex vivo cell engineering strategy with prolific protein production in vivo. That's done by taking B cells, using CRISPR-Cas, followed by homology direct repair to do the engineering. That engineered B cell is then differentiated into a long-lived plasma cell. This is one of the most biosynthetically capable proteins in your body. It produces thousands of proteins per cell per second. That cell is collected and then can be infused directly into patients, again, without any preconditioning. It homes very quickly to the bone marrow, which establishes itself in bone marrow niches. You all have in your blood right now anti-measles antibody, because as children you either got the vaccine or you had measles. Those plasma cells will continue to produce that anti-measles antibody for the rest of your life. So we have an ex vivo engineered cellular factory with in vivo therapeutic protein production. We can produce any class of proteins, therefore we can potentially treat multiple types of applications. We've engineered these cells to make things as small as 39 amino acid and GLP-1R agonist peptides. Plasma cell has no problem making and secreting a peptide. We've produced them to make a variety of different enzymes and factors. I'll show you 2 examples of that. They can make complex proteins, multiple targeted bispecifics, complex fusion proteins. Our 102 medicine actually is a complex fusion protein. And of course, you'd expect them to produce antibodies, but not only the IgGs, but the more complex antibodies, the IgAs, the IgMs. And there are no currently approved medicines based on IgM or IgA today because they're very difficult to manufacture. Plasma cells make them every day with no problem at all. To make a BCM, and to have that predictability and durability, there's things we focus on. First is precision engineering. So shown in that Venn diagram is some of the data we obtained to document precision. So we're using CRISPR-Cas. We put our transgenes into the gene called CCR5. It's a safe harbor locus. You can be deleted for CCR5 and you're perfectly healthy. So that greatly eliminates the concern for insertional immunogenesis. Transgene, same location every time, every product. Copy number cannot get over 2. You only have 1 copy of chromosome for each parent, so our copy number never exceeds 2. That predictability is then translated for predictable protein production. Same location, same copy number, production after production after production. In the center panel is information we obtained from mice a year or two ago. So you don't have to worry about the details, but the red line is what happens when you take a human plasma cell, put it in an immune deficient mouse. You're looking at production over time. The blue line is the similar approach where you now put that human plasma cell into a mouse which has a human immune system, so it's a Hu34 mouse. What you see is better engraftment and better persistence with an intact immune system. Again, very distinct from HSC gene therapy or CAR T cell therapy. We have to get rid of the endogenous immune system to get good engraftment. No, plasma cells want that intact immune system to get ideal engraftment. And you can follow the production of these cells out for over a year in mice. Here's data where we validated this in our non-human primate model. So we worked with a group at the NIH. They are able to apheresis a rhesus monkey. We take rhesus monkey B cells. We turn those rhesus monkey B cells into antibody-secreting plasma cells. We then radiolabel those cells and give them back to the animals. Simple IV infusion, no chemo, no conditioning of any kind. They very rapidly home to and engraft in hematopoietic niches, such as the bone marrow and spleen. Top left, PET image. Next to it is the CT overlay. So because this is a PET emitter, you can go inside the bone with the CT, and you can see exactly where your plasma cell landed. An example is shown there in the spine. We see cells in the spine, which is a major reservoir for your hematopoiesis, within an hour of infusion, and it's still there and increases a little over time, 6 days later. We've also been able to redose these animals. Bottom panel shows dosing. And the next day, we again do the CT imaging. You can see the entire bone system and spleen is lit up. This isotope has a 3-day half-life. It's gone completely within a couple weeks. We retreated that animal 3 months later with a new batch of cells. We get the exact same biodistribution and the exact same percentage of engraftment. So we know that 20% of the input material goes to bone marrow and spleen, and that's a reproducible number with an infusion that's 3 months apart. Okay, so let me tell you about our first program, B101. This is for Hemophilia B. Hemophilia B is a large and growing market. Standard of care is prophylaxis therapy, requiring 26 to 52 infusions per year for life. Despite the ready access to prophylaxis, 25% of severe hemophiliacs don't do routine prophylaxis, and 80% of moderate hemophiliacs don't. Even in the best case of prophylaxis, you still have the peaks and troughs where you infuse, half-life, you decline, and it goes down. This is actually a relatively expensive drug. We're talking about patients spending $750,000 per year for life. Because of that peak and trough, there's still significant unmet need for Hem B. 70% of the patients have multiple bleeds per year. That leads to joint damage. That leads to chronic pain. There are over 40,000 patients worldwide. There's greater than 5,000 patients with Hem B in the US. Sales worldwide last year for prophylaxis were over $2 billion, and that's expected to continue to increase over the next decade. So delivering this protein by a B cell, which engrafts in the marrow, constitutively produces factor IX, we believe will have a best-in-class TTP. If you look on the left, the attributes are distinct and cumulative when you compare prophylaxis versus AAV gene therapy. So our B101, we will have constant factor IX production, which will be durable. We're aiming to dose once or at most twice a year. So that durability is key difference between the current therapies where you're going to get the peaks and troughs. Compared to AAV gene therapy, we can redose, we can titrate. If a patient has a certain level but they want to change their activity, we can give them another dose. That redosability is critical for the adoption of the medicine. And we can treat children. Because with AAV gene therapy, you can't treat kids because as your liver grows, you can't redose them because once dosed, you are then immunized. So we believe we can very quickly have the best-in-class TTP for Hem B. We have a phase 1 trial, which is underway. We dosed our first patient in July. These are for patients with moderately severe to severe hemophilia B. They have to be in relatively good condition and have already received factor. That dose escalation will finish next year, where we then go on to our dose escalation with that number. We will then confirm that dose, go into kids, and then be able to retreat patients starting next year. So this is a big year for us. It's the year we turn our vision and our cutting-edge science into clinical data for a best-in-class profile on behalf of patients with hemophilia B. Let me tell you the pipeline, starting with, again, our next program, which is B102 for hypophosphatasia. Hypophosphatasia is a defect in alkaline phosphatase. In the severe forms, you don't form bone. You also have a variety of other sequelae and can range in various severity, particularly actually in the neonates with severe disease. There's only one drug to treat HPP. It's called Strensiq, marketed by AstraZeneca. It requires 3 to 6 injections per week, 3 to 6 injections per week for life. Those are painful injections. A lot of injection site reactions, therefore there's a high discontinuation rate. It's only approved, the label's only approved for the patients who are diagnosed as juveniles, and despite that, being able to only treat 2,000 of the estimated 100,000 patients with alkaline phosphatase deficiency, it's already a billion-dollar drug. Here's some of the data that left to our development candidate. On the top's just a diagram how you can actually trick an osteoblast cell line into forming bone, which you can then stain for calcium. It stains red. If you put in an inhibitor which accumulates in HPP patients, you block the calcium deposit. So in the photomicrographs there, you can see that beige staining means you're not forming bone. If you add the drug Strensiq, either 50 or 100 nanograms per mil, you can actually see activity, bone formation, staining red at 100 nanograms per mil. We purified the protein from our B cells to the same dosing. We saw activity at both 50 and 100, so it's a very potent drug. Put those into mice, and you can see, again, post-engraftment constitutive production over time, and we know that molecule goes to the bone and actually is active in the bone. So therefore, we think for hypophosphatasia, this is clearly will be the best in-class TTP. We're looking for yearly infusions, which again will produce that constant level of alkaline phosphatase. This addresses, again, the underlying need in HPP patients. It's targeted to the bone. It will almost certainly avoid the painful repetitive injections. And one of the things also is that it's a very immunogenic medicine because of those repeat subcutaneous injections. So we are looking forward to advancing that into the clinic with an IND next year. To finish up, I want to tell you like 3 other applications potentially. I told you we can produce any type of protein. Here's 3 examples of that. Starting on the left, we have B cells producing an enzyme replacement therapy type approach for lysosomal storage disease. This specific LSD is Niemann-Pick disease. We have a mouse model, which is a knockout for the protein in Niemann-Pick disease. It accumulates metabolic byproducts in the liver. They can stain green. If you take those mice and you give them the B cell making the enzyme replacement therapy, you completely clear that toxic buildup in the liver. We've also been able to show we can do this in the CNS as well. In the middle panel is B cells making a bispecific T cell engager. This essentially is a mimic for the Amgen drug Blincyto. That drug has a half-life of 4 hours. Patients who are getting treated for leukemia with Blincyto have to wear a backpack such that they get continuous infusion for a month. We're able to give a single dose and cure that tumor. And of course, you'd expect us to make antibodies. Here's an example where a mouse is given B cells making an anti-RSV antibody. Challenge that mouse with RSV, you get no virus production. So therefore, I hope you can now appreciate the power of B cell medicines. So B-Bio, a multi-program clinical stage company with data now clinically coming in. We believe this is a groundbreaking best-class approach to the delivery of in vivo biologics. Rapid concept to IND, first program, less than 2.5 years. Our BCOM9 trial for Hem B patients is up and running with brisk enrollment. 102, game changer really for HPP, DC this year, IND expected next year. So B-Bio, we are well capitalized, multiple program, clinical stage company, very excited for the patients that we were born to serve, eyes locked on the patient, every decision always. So thank you.
Thank you, Biopharma.